One of your classmates brought it to my attention that on question number 2, there were really 2 incorrect answers. Both D and E were incorrect. E was really totally incorrect. An absorption spectrum isn't the same as an action spectrum. They're very different things. But the question prior to that said that chlorophyll has 2 functional parts. That's true. Has a long hydrophobic tail. That's true. And then it says it has a complex ring structure with iron in it. I read that several times myself. I intended to put magnesium in there, but I put iron in there, and that is incorrect as well. So if you miss that question, I will change that one. Quiz Monday will be over section 13.4, which is in the previous chapter that we covered on DNA. That's some applications of DNA to biotechnology. And I thought that it would be a good time to give you a quiz on that because that directly relates to stuff you're doing in lab. So you'll learn a little bit about, if you haven't already, polymerase chain reaction. You should already know about electrophoresis, restriction endonucleases. So a lot of this should be reviewed for you, but just those 4 pages, 261 through 265. And then anything is there going to be some background information next Monday that you'll need for lab?
Just a little. Not a lot. It's just doing the transformation associated with people.
Okay.
It's just a difference in selection.
Okay. We'll talk about that. Okay, we were in the midst of, any questions? We were in the midst of talking about mutation and the fact that there are typically 2 general categories of mutations. The spontaneous mutations occur as a result of errors in DNA replication. Nothing's perfect. DNA replication comes awful close, particularly because it has the built-in proofreading mechanisms associated with it. But mutations do creep through. And most typically, they involve just the substitution of one nucleotide for another. We call that a point mutation. We then talked about the effect that point mutation or base pair substitution could have on the phenotype. And there may be no effect at all. That's probably most common. We can have an altered genetic code, which results in a slightly altered sequence of amino acids in the polypeptide. Or a nonsense mutation can be pretty serious because that turns a codon for an amino acid into a stop codon, and that would then terminate polypeptide synthesis. So you'd make a very short polypeptide chain, which obviously wouldn't fold up properly and give you the function that you need to have. I'd like to go back to this one for just a little bit because this missense mutation is the basis for a very well-known and studied genetic disease. Did you talk about sickle cell last semester? Okay, so you already know a lot of this background information. Sickle cell anemia is the result of a spontaneous missense mutation. And the result of this mutation is an altered hemoglobin molecule. Now remember what you know about hemoglobin. It is a functional protein. Its role is to carry oxygen. And it consists of 4 polypeptide chains, so it's got quaternary structure. 2 alpha chains, which are identical to one another, and 2 beta chains, which are identical to one another. The effect of having this particular gene defect is going to depend upon a couple of things. Number one is how many copies of that gene are defective. Remember that you have pairs of genes for each trait, since you are a diploid organism, one from mom and one from dad. And the consequences of having this particular defect vary depending upon whether you have 2 altered copies of the gene, 1 altered copy of the gene, or no altered copies of the gene. And in addition, there are some environmental consequences associated that affect this. So, on the left is our wild type hemoglobin DNA. And on the right is our mutant hemoglobin DNA. So you can see that there is a single base pair substitution. A T has been changed into an A because of an error in DNA replication, more than likely. That affects this particular codon right here, and the GAA is turned into GUA. And that results in, instead of glutamic acid being coded for by the message, a new amino acid, valine. So we have a single substitution of valine for glutamic acid. There are some consequences to that. And one of the biggest consequences is that this mutation, which affects 1 out of 146 amino acids in the polypeptide chain, can result in a change in the shape of the normal red blood cells to this sickle shape. And you can phenotypically see this under the microscope. Okay? One of the things that really tends to stimulate these cells going from this shape to this shape is if the environment becomes a little bit acidic. Okay? So even if you have sickle cell disease, sometimes your cells will look like this, but then they can shift into this shape depending upon how much acid is present. So acid promotes cell sickling. Okay? So if we use a capital N to represent the normal hemoglobin gene, and a small case N to represent the mutated one. Okay? So here's our normal hemoglobin, capital N, mutated hemoglobin, small case N. Okay? So there are 3 phenotypes that you can have. There are 3 genotypes you can have. Homozygous dominant, heterozygous, homozygous recessive. What are the consequences if you are this particular genotype? What condition do you have? You have sickle cell anemia, okay? And what happens when you have sickle cell anemia is that it is very common for your blood cells, virtually all of them, to assume this sickled shape, particularly if conditions get a little bit acidic in your blood. And one of the things that can promote sickle cell crisis is exercise. Okay? So if you exert yourself at all, that tends to cause those sickle cells to sickle. Is that a problem? Can you see it being a problem that these cells have this shape as opposed to this shape? How come? What's the problem with being sickle shaped? Okay, it might affect your ability to carry oxygen, that's true. Okay. Yeah, they get clogged in the capillaries, okay? I don't know if you've ever seen before pictures of the circulatory system, but by the time the blood cells get down into the little tiny capillaries, the capillary is barely big enough to contain one blood cell. And so those ragged edges and stuff, those sickled cells, get trapped in the capillaries, and it basically clogs up circulation. And that's not good. It's painful. This also tends to facilitate a breakdown of these red blood cells. You might know why these cells might be broken down more than regular shaped blood cells.
Because the body recognizes them as like bad.
Yeah, the body recognizes them as bad. And particularly your white blood cells, the phagocytic cells, see these sickle cells and they go, they don't belong here. I'm going to break them down. These sickle cells also tend to accumulate in your spleen. So one of the things that happens if you suffer from sickle cell anemia is your spleen becomes very enlarged. Well, look at this list of consequences of breakdown of red blood cells, clumping, accumulation in the spleen. Physical weakness, anemia, heart failure, pain and fever, brain damage, damage to other organs, spleen damage. And then they have their subsequent effects too. Kidney failure, rheumatism, pneumonia, paralysis, impaired mental function. That's a nasty set of conditions, right? And so you would think, people who have this particular trait have really lost the genetic lottery, and in some respects they have. This particular disease is oftentimes lethal. So that's pretty amazing when you think about it, is the fact that one amino acid change, because of one change in a nucleotide, can result in lethality. Well, it's kind of interesting. Normally, natural selection selects against these lethal genes. And yet, when you look at some parts of the world, the prevalence of the sickle cell gene in the whole population is very high. And in fact, it's much higher than we would have expected it to be based upon the fact that it's such a damaging gene. In what parts of the world is sickle cell gene frequency higher than we would normally expect? In parts of Africa, yeah. This map shows you, and it's not just Africa, there's Middle East, there's India. This shows you a breakdown of kind of the frequency of the sickle cell allele. And you can see red is higher frequency, white is low frequency. And there are some real hot spots around these particular areas where we find large frequencies of this particular gene. Okay? This is much higher than you would expect normally for a gene that's lethal. So what do these areas have in common? These are areas where there is a high incidence of malaria. Okay? What do you know about malaria?
It's passed by mosquitoes.
Yeah, it's transmitted by mosquitoes, and the mosquitoes carry a little protozoan parasite. And when they feed on you, the parasite gets into your blood. The parasites actually enter your red blood cells, and they replicate in the blood cells. And then the blood cells burst and release more parasites, and they infect more blood cells. And so you get all kinds of red blood cell lysis, fever. It can kill you. So why is the incidence of this lethal gene so much higher in these particular regions where malaria tends to occur?
The shape of the sickle cell doesn't allow for the protozoa to get in and multiply.
Okay, it has something to do with the fact that if you have this particular genotype, if you are heterozygous, you have what is referred to as heterozygote advantage. When do you have that advantage? In the presence of malaria. Individuals with this particular genotype are more resistant to malaria than either these individuals or these individuals. Okay? In the absence of malaria, is there a real consequence to having one good gene and one bad gene? No, there really isn't. Okay? You seem to be okay in terms of normal hemoglobin function. But in the presence of malaria, an advantage kicks in. Okay? So what's the advantage? Well, the advantage is that if you are heterozygote, approximately half of your cells are potentially sickled in the presence of acid. Half are normal, okay? Well, why would cells become acidic? Because during the infectious process, those parasites get into your red blood cells and they metabolize there. And when they metabolize, they produce acidic products. And so the cells sickle when the red blood cells have been infected by parasites. How does your body respond? When it sees those sickled cells, what does it do? The white blood cells recognize them and destroy them. Okay? So basically having this mutant allele gives your body a way to recognize when blood cells have become infected. And therefore you can eliminate those particular red blood cells. Okay? You do not have that protection in either of these situations. Only in the heterozygote state do you have that heterozygote advantage. The video you're going to see in lab next week talks about sickle cell, and it's got a nice little movie that shows the parasites actually invading red blood cells. It shows them sickling them, and then in that sickled state, they're kind of marked for destruction by your body's immune system. So a single gene defect has really influenced evolution, particularly human evolution, in this interesting relationship between malaria and the sickle cell disease. Okay, we also talked about induced mutation a little bit. These are caused by environmental damage. We also mentioned earlier the ability of the cell to correct some of these induced mutations. These excision repair enzymes, those endonucleases that can cut out the TT dimers, which form in response to ultraviolet radiation. And we mentioned the genetic disease, xeroderma pigmentosa, where individuals have a defect in these repair enzymes. It's really important for cells to be able to repair these induced mutations, because if mutations persist, that oftentimes is one of the initial events that occurs that turns a cell from a normal cell into a cancerous cell. And so the relationship between cancer and mutation is pretty strong. There are also various chemicals that can cause induced mutation. I think I mentioned earlier on that some of these chemicals actually are structurally similar to nucleotides and can actually insert themselves into the DNA, or can cause nucleotides to be deleted from the DNA. That causes an interesting problem. Previously, in cases like sickle cell anemia, we're talking about a substitution of a nucleotide, changing one to another. Again, most likely the result of a spontaneous mutation. However, many of these induced mutations cause either the addition or deletion of a nucleotide, and that gives us a little bit different scenario. That results in a type of mutation called a frame shift mutation. The result of that frameshift mutation can be either missense or nonsense, just like it was in a point mutation, but the mechanism is a little bit different. I'll explain this in just a second, but the worst kind of frameshift mutation is one that involves the addition or deletion of 1 or 2 nucleotides. 3 is not nearly as serious. And if the mutation occurs at the beginning of a gene, it's much more serious than it happens at the end of a gene. So, here's our wild type. Here are our code words in the message, and here is the wild type protein. What if you were exposed to a chemical that essentially resulted in the insertion of an additional nucleotide into this message? I'm sorry, into this DNA. And it resulted in a message being formed that had an extra U. Here is the normal reading frame for that message. Here's one codon, here's another, here's another, here's another, here's another. Here's the new reading frame. Here's a codon, but now this is the next codon, okay? UAA, not AAG. We've shifted the reading frame by adding that one additional nucleotide. And UAA is a stop codon. Okay? So immediately, this message now only contains the information for a single amino acid. What if instead the mutation resulted in the deletion of a nucleotide, this U right here? What effect would that have? This codon would be fine, this codon would be fine, but this next one now, which would have been UUU, the U is removed, so now it's UUG. So that codes for leucine as opposed to phenylalanine. From that point on, the reading frame has been shifted, so every other codon from that point on is going to be affected. So this will no longer be glycine, this will be alanine. So we'll change every subsequent code word by pulling out one nucleotide from the reading frame. This gives us extensive missense, okay? Because everything basically from this point on is wrong. What if we delete 3 nucleotides? Then that really only affects one codon. And we haven't altered the reading frame. We're just basically missing one amino acid in this particular sequence. So by deleting an AA and a G, we do miss lysine now, but then all these other code words down here are still in the proper reading frame. And so the protein would be methionine, phenylalanine, glycine, as opposed to methionine, lysine, phenylalanine, glycine. So I think you can see why inserting or deleting 1 or 2 nucleotides causes frame shift. Inserting or deleting 3 nucleotides only affects that single code word and that single amino acid. Hi, Jenna.
Can it take out part of one and part of another as a 3?
Part of one and part of the other. I'm not sure what you mean.
Like take 2 from one and 1 from another?
Oh,
Is that
yeah, it could. Okay, so you're right. Okay, so if it was, 3 here, yeah, that would affect everything. Okay. Good point. And again, if this happens toward the beginning and it causes a frame shift, that is more damaging than if it happens at the end of the message, because that would affect fewer amino acids. Your cell's DNA is under constant assault. And there are all kinds of things in the environment that can cause mutation. Fortunately, your cells have pretty good repair mechanisms that catch most of those mutations and mean they don't make it on and aren't passed on to other cells. If mutation occurs in a somatic cell, don't know what that term means, somatic? It means a body cell as opposed to a germ cell. Then that mutation could be passed on to your other body cells. Mutations that occur in germ cells, those that produce sperm and egg, have the ability to then be passed on to future generations. And so some genetic alterations are inheritable. Because genetic alterations are oftentimes the first step in cancer, some cancers are inheritable. And the tendency to get a cancer can run in a particular family. The relationship between carcinogens and mutagens is pretty strong. A mutagen is a substance that causes mutation. A carcinogen is a substance that causes cells to become cancer. Okay? About 90% of all known carcinogens are mutagens. So there's a high relationship between those two terms. One of the things that we're always concerned about is are we being exposed to substances that are potential mutagens and or carcinogens? And that's particularly true in the food we eat. If you look at the labels of food, especially processed food in the grocery store, there's a whole long list of chemicals that are in most foods, and the purpose of those chemicals is to serve as food preservatives. Are those safe for us? Are those particular compounds potentially cancer-causing compounds? That's always a concern. Well, testing to see whether something is really a carcinogen or not is incredibly time-consuming, expensive, and it's a big deal. Most of the time, it involves laboratory studies of animals like mice and exposing populations of mice to chemicals like food additives and seeing if they develop tumors. But just because something develops a tumor in a mouse doesn't mean it's carcinogenic in a human. Not necessarily. And ethically, we don't take humans and expose them to potential carcinogens and see if they will develop cancer. So developing models that can kind of predict whether compounds are potentially cancer-causing is a big deal in research. Well, a test was developed years ago called the Ames test that tried to develop a simpler model for testing things as being potential carcinogens. And the Ames test doesn't use rats. It uses bacteria. And the purpose of the test is to expose the bacteria to the chemical that you're testing, maybe it's a new food additive, and see if it can cause a mutation in that bacteria. And if it causes a mutation, a red flag is raised. Why? Because so many of those mutagens are carcinogenic as well. And this would suggest, man, we better follow up and test that compound more to see if it's potentially cancer-causing or not. If it doesn't cause mutation, then there's very little likelihood that it's also cancer-causing. I'm not going to get into how these bacteria, how we can look at bacterial plates and determine whether they have mutations. But it really has to go basically with how they grow on certain types of agar media. But there's this relatively simple and dirty test, the Ames test, where you can put some bacteria on a plate, expose them to a chemical, and easily determine whether a mutation has occurred. And if a mutation has occurred, that's a red flag saying that's a potential carcinogen. We really need to do more animal testing to kind of nail that down. I've made references throughout this last couple of weeks to the importance of organisms being able to regulate the expression of genes. What two things have to happen in order for a gene to be expressed? Genes have to be transcribed, and they have to be translated. There are a lot of opportunities in both prokaryotes and especially eukaryotic cells to regulate gene expression and to see how that happens, when that happens, and where that happens again. One of the things that's really fascinating about the regulation of gene expression in both prokaryotes and eukaryotes is that those organisms can really alter their gene expression in response to changes in their environment. We're going to begin this discussion in a couple of minutes, concentrating on prokaryotes and looking at how they respond to changes of different foods in their environment. But there are all kinds of other environmental signals that can cause changes in gene expression. What do you suppose a big one is in plants? What's an environmental thing that can cause change of gene expression in plants? Temperature?
Sunlight.
Light? Absolutely.
In the water?
Potentially water. Okay. In humans, it might be hormones. In insects, it is definitely hormones. So there are all kinds of environmental cues that can allow an organism to respond to changes in its environment at the genetic level. In multicellular eukaryotes, then we also have changes in gene expression involved in development of the organism. And changes in gene expression are responsible for the development of different cell types within a multicellular organism. Remember that all of your cells have within their nucleus the genes to make an entire organism. In some cells, certain genes are turned off, and other cells, certain genes are turned on. One of the things that you're going to see, and it's going to kind of blow your mind a little bit, but there are all kinds of interactions that take place between different molecules that help to regulate gene expression. There are protein-DNA interactions. There are RNA-DNA interactions. There are RNA-RNA interactions. And all of these play an important role in regulation of gene expression. This is the one that's kind of emerging right now as a really hot area of study, and we'll talk a little bit about that one next week. See a little short video about RNAi, or RNA interference. This is a very productive area of study right now and has some real promise for treating some pretty serious medical conditions. So let's start simple. Let's start out with bacteria, and let's look at the response that bacteria can have to changes in their environment by regulating transcription of genes. Bacteria, like all other cells, are subject to natural selection, and natural selection favors economy. Don't use resources if you don't need to. Only produce things that you need to. Don't waste energy transcribing genes, translating them into proteins if you don't need that protein. Well, one way cells can regulate metabolism is by regulating the production of enzymes by feedback inhibition. Another way they can do it is by regulating genes themselves. So there's kind of two basic levels under which organisms can kind of control their metabolism. through regulation of enzyme activity or through regulation of gene expression. Regulation of enzyme activity, particularly by feedback inhibition, is much faster, but doesn't save as much energy as regulation by gene transcription. So let's give you an example here. Okay, here's a metabolic pathway. Here's some precursor molecule, and several enzymes convert one thing to another, and this results in the synthesis of an amino acid tryptophan. Okay? This is one of the 20 amino acids. Everybody needs tryptophan to make polypeptide chains. Okay? This amino acid is going to have to be hooked onto a tRNA at some point in time and incorporated into a polypeptide chain during translation. If you have a lot of tryptophan in your diet, do you want to waste your resources turning this precursor into more tryptophan? Probably not. Particularly if that precursor could be turned into something else you need. Remember that metabolic pathways are oftentimes branched, and one thing can go multiple directions depending upon the cell's need. So if you temporarily have a lot of tryptophan, you could immediately shut down this pathway because tryptophan is a feedback inhibitor of this particular enzyme. It binds to the enzyme, probably an allosteric site, changes its shape, and results in this enzyme not being active, so that the precursor isn't converted into tryptophan. Maybe the precursor is used to something else. This can happen almost instantaneously. Okay? But does this save you a lot of energy? No. You're still making these enzymes, even though you're not using them. So this is good for kind of short-term response, okay? You get a little spike in tryptophan in your environment, and therefore you shut down temporarily the activity of these enzymes, and you don't make more of it. But what if tryptophan was kind of basically present all the time? You had a permanent source of tryptophan. Then there are mechanisms in bacteria also that allow that substance to actually influence gene expression and determine whether these enzymes are even made or not. That's a whole different level of regulation. That process takes a little bit longer to play itself out. But it saves a lot of energy because you're not even transcribing these genes to make these enzymes. So this is kind of a slower level of control, if you like, but it saves you a lot more resources in terms of energy. So this is the one that we want to concentrate on a little bit, is how can substances in the environment actually affect gene expression, turn on or turn off genes, as opposed to over here, we're turning on and turning off enzymes. Okay? So here we're working at the transcriptional level of the central dogma. Over here, we're looking at enzyme activity being regulated. Well, there's a wonderful model that was proposed way back in the dark ages of 1961 by two French researchers, Jacob and Monod, at the Pasteur Institute. And they suggested that bacteria have this really cool mechanism that allows them to regulate gene expression, and they called that particular mechanism the operon model. And the key here is that this thing called the operon consists of some various elements. A repressor gene, an operator gene, and a structural gene. Let's take each one for just a second. Look at it in a little more detail. The repressor gene is a sequence in the DNA. It's always active. And it codes for a protein called the repressor protein. The repressor protein can occur in two states or conformations. One shape is active and one shape is inactive. What's going to cause this molecule to change shape from one state to another? Other molecules bind to the repressor. Okay, so if my hand is the repressor protein, and let's say in this shape it is the active shape. But if another molecule, this piece of chalk, binds to it, that changes the shape of that repressor protein. And now that might be inactive. Release that molecule and it becomes active again. Bind that molecule and it becomes inactive. So this protein can alter between these two states or conformations of active and inactive, depending upon what binds to the repressor protein. Second element of the operon is a region called the operator. The operator is actually a part of the gene sequence that we've already heard about called the promoter. Remember what role the promoter plays in transcription? What binds the promoter and allows transcription of a gene into a message? RNA polymerase. Remember RNA polymerase is the molecule that binds to that promoter, and the transcription factors help the position of the promoter. And once it's bound to the promoter, it can then move down the message and transcribe it. So this is a part of the promoter. It's a segment of the promoter. And basically, it can serve as a roadblock. Like a bunch of orange traffic cones. And if that operator is blocked, no transcription occurs. So if your car is the RNA polymerase and your road is the gene it's going to transcribe, if there are a bunch of traffic cones that block you from going down the street, you're not going to get anywhere. But if the traffic cones are removed, then you can drive down the street. You can transcribe the gene. Well, what determines whether this operator is blocked or unblocked? The repressor protein does. The repressor proteins are the traffic cones. And if the repressor proteins are in the active state, they bind to the operator and block it, and transcription doesn't occur. If they're in the inactive state, then they can't bind to the operator, and therefore polymerase can just move down and copy the gene. Make sense so far? Fortunately, it's a bit more complicated. Okay, third element, structural genes. Structural genes code for polypeptides. So, in this pathway right here that involves tryptophan synthesis, there are 5 enzymes that are involved in this pathway to make tryptophan. Each one of those is encoded by a structural gene. If I don't want to make tryptophan, I don't want to make any of these enzymes. It's not like I say, well, I'll make 1 and 3, but not make 2 and 4. Either make them all because they're all involved in the same process, or don't make any at all. So all of these, basically, genes are under control of a single element, this operator. And if the operator is blocked, we don't transcribe any of those 5 structural genes. If the operator is unblocked, we transcribe them all. So this allows groups of genes that are related to be under the control of kind of a single control element. Okay. There are kind of 2 categories of operons. There are reducible operons and there are repressible operons. Inducible operons tend to regulate enzymes that are involved in catabolic pathways. Repressible enzymes, repressible operon, are involved in regulating anabolic pathways. Remember, catabolic, we're breaking stuff down. Anabolic, we're building it up. We're going to use as an example of our repressible operon the anabolic pathway of tryptophan synthesis, what have we just been talking about. For our inducible operon, we're going to look at the breakdown of the sugar lactose into its 2 monosaccharides, glucose and galactose. The operon that's involved in regulation of lactose metabolism is called the lac operon. And the operon that's involved in the synthesis of tryptophan is called the trip operon. We're going to look at the lac and the trip operons, kind of contrasting examples of these 2 situations. So if you haven't been reading, I would suggest you start working on chapter 15, and we will continue to move through this stuff tomorrow and talk about the 2 operons. If you don't have your exam with you and I misgraded that question number 2, just bring it by to me sometime or give it to Dr. Morgan in lab. I would have to look and see what. Send me an email to remind me, and I will give you some scenarios. Okay?
Should I put it in this pile?
Thanks, Ann.
Yeah, just put it, that's
Hi.
fine.
I have a question about this. Were you supposed to use, I remember I think we turned it in, had all the plates and the numbers of counties. We turned that in, but I thought we got it back.
Thank you.
Actually, I
Yeah, I mean, you were supposed to turn this in,
Really?
yeah,
I showed you that wasn't really clear. I used,
last, 2 weeks ago.
Really? Oh my god.
yeah,
Yep.
I think so.
Yeah, so that one's beginning of next week's lab.
Is there any other kind of other extra optional stuff I can do? Like, can I still do notes for that one video, the secrets of
Yes, you can.